Hey, how's it going? Today we're going to be setting up some things for mitosis and meiosis, and we'll talk a little bit about the introduction to genetics. So for this week, I just wanted to wrap it up, some ideas for mitosis and meiosis. What you should be able to do is know the steps, what happens, and why is it important. So mitosis and meiosis, what are their goals, what happens, and I'll preview that a little bit today. And also, for intro to genetics, what you should be able to do at the end of this thing is the simple Punnett squares. We'll practice these in class, but we'll hit some of the vocabulary. We'll talk a little bit about Mendel. We'll talk a little bit about DNA and why these two are linked. So the idea of genetics is related to meiosis, which is why these two are discussed usually together. So we'll start off, we'll do some quick reminders about mitosis. I'll jump into meiosis, I'll do a little compare and contrast. Then I'll hit intro to genetics, a little bit about Punnett squares and Mendel. Quick review of mitosis. Remembering mitosis, the goal is to make diploid identical cells. Diploid, di meaning two, so these are two end cells, meaning they have two copies. One from the mom, one from the dad. We're making diploid identical cells. And what are the stages? When you can remember IPP, MATSI, I meaning interphase. So here's where we have a division. Interphase is your general growth, your everyday run-of-the-mill cell. We talked about some of the regulation mechanisms for how you enter from interphase into prophase. So we have things like the cyclins that are important here. So that's how you can get into the actual mitosis. So once we're in actual mitosis, what we have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So through these steps, we have arrangements of DNA and chromosomes that we're coiling up. Some reminders about DNA. Remember, we have DNA, which is our double-stranded. We have chromatin. We have chromosomes. And we have sister chromatids. So keep those all in mind when you're remembering the steps. So those are kind of the quick review of what is in mitosis. I'll quickly go over the differences between mitosis and meiosis next. So take a moment, flip through your book. If you're not sure what the steps are in mitosis, make sure you can at least draw these steps, identify them, talk about why they're important, and know the different kind of levels of DNA as DNA changes throughout the steps of mitosis. Talk about meiosis. What's really important about meiosis compared to mitosis? We're going to be making haploid, one copy, and these are different cells. So you'll see this for spermatogenesis, making sperm, oogenesis, making eggs. So what's going on in meiosis that we're making different cells? What makes it so important? These steps of recombination. Recombination is your mixing up genes. And so where this happens is part of metaphase. So in metaphase 1, your chromosomes are attached as such. These are analogous chromosomes, or homologous, meaning they code for the same set of genes. So homologous chromosomes they'll code for the same set of genes. So same set of genes. So for example, hair color, eye color, so forth. We have recombination, meaning that these limbs will then reattach. So in our next step, what we'll have is we'll have this crossover event. So this one will be green with a little tip of black, and this one will be black with a little tip of green. This creates a new combination that was not present before. So we have a new combination, and that's really what's important about meiosis. We're making new combinations, and we're also halving the number of chromosomes. So if you started out with 46 chromosomes, this is your diploid number, you're able to go to 23 as your haploid number. Some equations will have you go back and forth between diploid and haploid. Can you identify the number of chromosomes? Can you identify the number of chromatids? So really quickly, meiosis recombination, a couple other steps with this. We are going from diploid to haploid. So because of that, we have 2 cellular divisions. Cell divisions, where instead of separating these, we now have tetrads. We need to separate. It's called a tetrad for 4 combinations, right? 4 parts. So that's meiosis. That would be a good time to make sure you can compare and contrast mitosis and meiosis. What's the difference? What do they have in common? What are their ultimate outcomes, and where do they happen? Those would be kind of a quick compare and contrast. So why do we talk about mitosis and meiosis before we get into genetics? It's because of the 2 laws that are predated and are caused by meiosis. So in meiosis, we have mechanisms that happen which influence genetics. Hey, how's it going? Remember, there was an old guy named Mendel. Mendel found a bunch of peas, he grew them, and he found these rules. So what are those rules that you want to remember? Law of independent assortment and the law of segregation. So these rules directly dictate what's going to happen in Punnett squares. The law of independent assortment says that genes sort independent of each other. Hair color should not influence eye color. Height should not influence other things. Next week we'll get into some of the complications for this, on why these rules don't always are true. For example, people with blonde hair tend to have blue eyes. But the idea is, in general, genes sort independent of each other. In the case of peas. So in peas, a green pea can be a wrinkled pea, can be a yellow pea, can be a small pea, can be a purple flower, can be a white flower. Law of segregation says you have random chance to get a copy. So you can apply these ideas into Punnett squares. This is our typical kind of monohybrid cross. You're just looking at one trait. That's why it's 4 squares. If it had been a 2 trait, we'd be looking for a 4x4 for each gamete. So law of independent assortment, we have a mom that is capital A, little a. We have a dad that is capital A, little a. They assort independently, so you could have an equal chance of getting an A, big A, or little a. Equal chance, 50-50. In this case only, special Mendelian genetics, 50% chance of each. Same thing over here. On the metaphase plate, what does that mean? You have an equal chance in meiosis side, you have an equal chance of getting the big A or the little a in your sperm or egg. We can then put these things together and come up with phenotype and genotype ratios. Recall, phenotype is physical, what you look like. And genotype is genetics, what your genes look like. So just a quick overview, we went from 2 heterozygotes, we cross them, we can figure out that we're going to have a mix. We've got kind of our first basic Punnett square going on. From here, you can then mate the ones inside. We can get F1s, F2s, parentals, and so forth. But just recall that some of these things are based on a real reason of meiosis. The reason why meiosis is taught is because it's the element behind all of genetics. So in summary, what did we touch on? In summary, we found that meiosis is the basis for genetics. We have 2 laws from Mendel. And we also talked about the difference between mitosis and meiosis, what the goals are for these 2. So that's kind of just a wrap-up of where we're going for genetic information. We're going to be talking about a lot about genes and how they form what you are in the next couple weeks, talk about central dogma. But just to get you started, hopefully that gave you some pointers, some places to start for when you're thinking about things like meiosis, mitosis, and a little bit about genetics.